Hello, my name is Michael Feig. I'm a professor in the Department of Biochemistry and Molecular Biology at MSU. If you want to find out if existing drugs could be suitable to act as a therapy against COVID-19, is basically to see if they can interact with these proteins on SARS-CoV-2. And in order to do that, so one can do this clinically, trial and error, basically in a blind fashion, and just try out different things. The smarter way is to use the protein structures and try to see if those drug molecules can interact, can bind strongly to those targets. And then if that is the case, then one can look at those drugs further and see if in the clinic they're actually effective. We're trying to build models that are good enough for these computational screening efforts to find out which of the drugs that are on the market, and also maybe new drugs that are being developed, which ones of those are most likely going to be candidates as a therapeutic against COVID-19. What we're doing are high accuracy models for proteins from SARS-CoV-2 for which there aren't any experimental structures available. So for quite a few, about maybe half of the proteins in SARS-CoV-2, we already have experimental structures. Experimentalists working very hard to solve those structures, but they have not been able to resolve all of the structures. And our specialty is high accuracy refinement, high accuracy modeling of structures for which there's no experimental structure available. So we have an established protocol that we've used in the past and that works pretty well, but we typically focus on smaller protein targets that are easier. And so the targets in SARS-CoV-2 are quite large. That presents computational challenges. Because of the urgency of COVID-19, we had to really push to get very quickly very many models at very high accuracy out. And I think that was probably the biggest challenge. So the biggest, how do we overcome the challenge, is basically find as much computer resources as we can. We use particular GPUs. We have some in the lab that we could use. We also got access to some of those GPU cards at national supercomputing centers, but it wasn't enough. Again, we wanted to generate models very quickly. HPCC was generously giving us access to some of their GPUs that we otherwise couldn't have gotten access to, and that accelerated our rate at which we could generate models. So in particular, we're collaborating with Professor Rami Amaro at UCSD and Professor Saima Khalid in the UK at the University of Southampton to essentially build eventually a complete model of the virus, in particular the viral envelope in high resolution. And so the challenge is now how the proteins come together to form the virus at high resolution. I think everybody should do what they're good at, and if they can contribute to help with COVID-19, that's great. And I think many of my colleagues are putting a lot of effort into using their specialty to contribute what they can, and I think every contribution is valuable and every effort is good. And I think many people are doing that already.